Outstanding, fantastic. Thank you everybody in the audience for joining today. Pleased to be with you here from my office at Fresno State, even though there's basically nobody here, some faculty that are teaching classes remotely but using the Fresno State infrastructure to do so. And so thank you again for joining. As Jill mentioned, I love participating in the OSHER talks. A couple of years ago, I think it was, I gave a talk on ancestry testing and interpreting ancestry testing data. And maybe some of you in the audience were there for that one as well. But today we're going to be talking about genetically modified organisms. All right. So the title is GMO Friend or Foe. And my goal as an educator for you today is not to tell you what my perspective is. I'm going to try to give you a balanced perspective on the pros and the cons of genetically modified organisms, and hopefully let you decide for yourselves whether to you a genetically modified organism is friend or foe. And if you do have any questions, because this is a webinar, you can't unmute yourselves and ask, but you are able to participate by using the chat and the Q&A buttons in Zoom if you have those available to you. So if I see something come in that's a question that really needs to be answered live, I'll stop and answer that question. Otherwise, at the end, we're also going to have a question and answer period if you'd like to interact with me then. As introduced, I'm a geneticist and an evolutionary biologist, and I've got the degrees, but I also want you to know a couple other things about me. One is that I have a YouTube channel where I post lots of research and science-related videos that I use for my classes. So if you're interested in genetics or evolution, if you go to that YouTube channel, youtube.com/c/Joseph Ross, you'll see the full list of my videos. Most of them are probably boring to you because they're class recordings. Virtual instruction this semester is really interesting, but I'm recording all of my classes and posting them for my students. You might skip those, but there's some nuggets of really interesting information hidden in there, including one of my recordings from the last OLLI talk I gave, the one on ancestry testing. You can also feel free to contact me at any point at my email address if you'd like to. Now, that's not really interesting. The most interesting things I can tell you for today about me are that I'm lactose tolerant. I love cheese. I love dairy. I love ice cream. And I also have wet earwax. And my goal by the end of the talk, if nothing else, is to at least help you understand why that is actually relevant to the concept of a genetically modified organism. So let's dive in. If we're going to talk about genetically modified organisms, it probably behooves me to give you a background refresher on the concept of genetics. So we're going to spend the first few minutes talking a little bit about DNA, which is a double helix, shown on the right here, in its physical form inside our cells. And by the way, the reason that I'm going to be looking down a lot as I talk to you is that this is where my tablet computer is, which is what I'm using to give this presentation. And if you see me looking off to the side over here, that's where I'm checking the chat. All of us humans have 23 pairs of chromosomes. So right now I'll circle one pair of chromosomes. Those are human chromosome 1. So we all have, in every one of the cells that makes up every one of our bodies, with rare exception, like blood cells, these chromosomes. And these chromosomes are genetic material. They're made up of what we call DNA. It's a chemical, and it has 4 letters. So think of the DNA chromosome as like a book. Each of our chromosomes is like a chapter. Each of the chromosomes has a specific sequence of letters. There are only 4 letters, A's, T's, G's, and C's, and a string, a linear string of those, like a linear string of words in a book, makes up each of our chromosomes. And it's the specific sequence of the letters. So here, for example, is part of the sequence of one of our chromosomes, A, T, G, G, A, G, C, T, etc. It's like a language. It's like English, but it's only got 4 letters instead of 23, or 26 rather. 23 chromosomes, 26 letters in the English language. Now, to give you a sense of the size of the human genome, which is the set of all of our chromosomes, all 26 pairs, if you add up all of the letters, if you count every single one of them, there are about 3 billion letters in the human genome, or the human book. The chromosomes, all of them together, provide the instructions for living things to live. And I'll explain a little bit more about that in just a second. So of the 3 billion A's, T's, G's, and C's that make up all of our chromosomes, let's focus on one, for example, the biggest chromosome in humans, chromosome 1. Each of those chromosomes, which is a physical object, it's a series of chemicals, the A's, T's, G's, and C's, it itself is 248 million letters long. And of course, all of this crams into each one of the cells, each one of the trillion or so cells that makes up a human body. So we have lots and lots and lots and lots of DNA in us that make up our chromosomes. And I point out here that this sequence, this short sequence, which is part of the gene called lactase, is found on our chromosome 2. So somewhere in each of our chromosomes, yours, mine, everybody else that's watching right now, we have a DNA sequence that is either this or very similar to this sequence of letters. And that sequence again is the gene that encodes a protein that's called lactase. And some of you might know what lactase does, and some of you might not. But before we get to what lactase is and why it's important for this story, I'll remind you that the importance of the sequence of letters in DNA, the sequence of A's, T's, G's, and C's, it seems random if you just look at it, but our cells know how to read that sequence of DNA, letters, and that sequence encodes a physical object called a protein. So you can kind of think about this using a cookbook analogy. DNA is like the cookbook that our cells use. It's the information that the cells use to create something like a meal. So the DNA is the sequence, and that sequence encodes proteins. Proteins are physical objects that do the actual work inside of our cells, helping cells divide, helping us live, helping cells produce and secrete insulin. All of the things that all of our cells have to do to keep us alive have to do with proteins. And each gene, in general, each region of DNA, encodes one protein. Lactase is important to us, or at least to some of us that are humans, because it is the protein that converts sugars that we consume in milk products into other sugars that our cells can use for energy. So to summarize, the specific sequence of letters in DNA encodes a specific 3-dimensional protein, a physical entity that is inside all of our cells, and those proteins are the things that do our cellular work. Now, in just a second, I'm going to show you something that's maybe a little bit better representation of that protein, because what I just showed you is 2-dimensional, but this is a 3-dimensional image. So this is a quick movie of that molecule being rotated in 3-dimensional space. So the DNA sequence of your lactase gene and my lactase gene and everybody's lactase gene produces in our cells a protein that looks something like this. And each one of those tiny little colored balls is one atom. So these are really tiny proteins, but they do important things. Now, where DNA comes into play and why the sequence of the DNA is important, that sequence of A's, T's, G's, and C's, is that every one of our cells, from fertilization, from sperm meets egg, where each one of us started as a single cell, a developing embryo, that cell has that full complement of chromosomes, all 46 of them, 2 times 23, 2 copies of 23 different chromosomes. that fits inside that single cell. But then, of course, all of us that are watching and participating right now are adults. We've got more than one cell. We've got trillions of cells that make up our entire bodies. So that means that when one cell replicates and becomes a two-cell embryo, the first thing that happens is that single cell has to duplicate all 3 billion of those nucleotide letters, the DNA letters, so that when that cell divides, each of the two resulting cells itself has that full set of DNA. And then when 2 cells become 4 cells, and 4 cells become 8 cells, and 8 cells become 16 cells, every time one cell divides and makes 2 in this exponential growth process, the DNA has to be copied so that every one of those cells has that full set of 3 billion letters that make up our 46 chromosomes. So, if you're still following along, great. If not, here's the time to tune back in for the key point. Our cells are not perfect at copying 3 billion letters every time a cell divides. I mean, imagine if you or I sat down, imagine we were in the Middle Ages, back in the monasteries where we had to copy books by hand. Imagine writing out a book that's 3 billion letters long. You'd probably make a mistake every once in a while. And that's exactly what happens in our cells as cell division occurs, as growth occurs. From the single cell to an infant to an adult, that increase in size is accompanied by millions of times when all of our chromosomes need to be copied so that every cell has a set of chromosomes. Those DNA sequence changes, the mistakes, maybe an A accidentally gets copied and is a G instead in one of these cell divisions. That change in the sequence of letters in the DNA is what we call a mutation. The DNA sequence occasionally, rarely, and randomly changes. It's very rare, but it's also random. And it's that randomness that makes all of us unique as humans. But I want to reiterate now and point out that these mistakes, these mutations, aren't necessarily bad. I think people tend to think, oh, mutation, and the first thing that comes to mind are people with 3 heads or 3 eyes or something like that. Most mutations don't have any effect. So a lot of DNA sequence changes that happen, these random mutations that happen during cell division, don't have any effect at all that we know of. Some mutations are bad, and some mutations can be good. And that's where we get back to the story of lactase, that enzyme I was talking to you about, the protein with a particular DNA sequence on chromosome 2 that we all have. And because I can't see you, I can't get a sense for the ethnic composition of the audience today, the 73 of you who are attending. But my guess is that there's a pretty wide diversity of humans in the audience. Most of you probably are lactose intolerant. That's my guess. And what that means is, as adults, what's normal, what's typical for a human adult is that they do not produce that protein, lactase, in adulthood. That gene on chromosome 2, that sequence of letters that encodes that protein, lactase, doesn't get turned on or doesn't stay on as we're adults. So normally, every infant, as hopefully all of you know, every infant drinks milk as the primary energy source for the first year, 2 years, whatever. Differs for some families. And so this enzyme lactase, that protein, that 3-dimensional structure we just saw, has a function. And what that function in all of our cells is, as infants, that enzyme takes sugars that are in milk. In this case, this is a structure called lactose. And it breaks that chemical in half, producing 2 different sugars, galactose and glucose. And those are sugars that our cells can use for energy. So we have to have this enzyme, lactase, this protein that breaks down milk sugars so that our bodies can use them. Right? That's infant humans. Adults. Normally, our cells stop producing that protein. So most human adults are what we call lactose intolerant. Because they don't produce that protein anymore, their cells don't, they can't convert milk sugars into the simple sugars that our cells can use. And that means that lactose, that enzyme, stays in our gut, in our intestines, where instead of our being able to use that sugar, bacteria use that sugar, lactose, and that's what tends to cause the bad things about lactose intolerance, like bloating and gas and so forth. It's the bacteria that are able to use the lactose sugar because we can't. So that is the typical situation. Now let's get to GMOs a little bit, although this is just an example. There are no genetically modified humans that are lactose tolerant, but at one specific spot on chromosome 2, where the lactose gene is located, there's a single letter, there's a C, in people that are lactose intolerant. And if you happen to be a person who at that same exact spot on the chromosome, in that exact spot in the DNA sequence, instead of a C, you have a T, you're lactose tolerant. As an adult, you basically have an infantile trait. That is, your body continues to produce that protein, lactase, into adulthood. That's weird. That's abnormal. That's not the way most human adults work. But those of us, including me, because I said at the start, I'm lactose tolerant, I'm a mutant. I have a T on that part of my chromosome where most humans have a C. And that single mutation, that one letter change in my genetic code, lets me digest milk sugars. And so I can enjoy all of the cheese and milk and dairy products and my ice cream as I want to. So those of you that are lactose tolerant that are watching, that trait that you have depends on genetic differences between you and people that are lactose intolerant. So a big point here is that simple genetic changes happen naturally. This is not genetic modification, by the way. These are mutations that happened naturally as cells were dividing and as the DNA was being copied thousands of years ago in the past in human history. These mutations arose, first happened, and they've been passed down from generation to generation ever since. And so those of us from the British Isles in Scandinavia, where I'm from, or where my history is from anyway, my lineage, that's one of the places on Earth where this mutation originally evolved. So a lot of people that descend from Scandinavia and the British Isles and Western Europe tend to be lactose tolerant for this reason. We all descend from that individual that first got this mutation, this single letter change. The other place on Earth where a lot of people are lactose tolerant is in the Middle East, where camel domestication started. And so that was a place where milk products were readily available, and some goats too, were readily available. And so that is another place on Earth where lactose tolerance arose. A different mutation, not this particular change from C to T, but a different one in that group of humans. So if you're not descended from Scandinavia or Western Europe or the Middle East, you're not very likely to be lactose tolerant as an adult. So this is an example of a naturally occurring genetic modification. Right? The concept we're talking about today is genetically modified organisms. This is a genetic modification. It's a single nucleotide change, a single letter change, and it changes a trait in humans. The main difference between what you probably consider to be a GMO, a genetically modified organism, and me is that you probably are envisioning, I'm just guessing, but what I would suppose is that you think of genetically modified organisms as scientists like me, but wearing a lab coat instead of a sweater, in the laboratory, tinkering with DNA and creating unholy combinations of organisms, taking a gene from one organism and putting it in another organism. And that's true. That's a different form of genetic modification. So we're going to talk about all of those sorts of concepts in the rest of our time together. I should definitely point out that you and I are related to each other somehow because we all descend from an ancestral human. We're all members of the same species. We are all Homo sapiens. And evidence of that is that any two humans, but if you take two humans at random, of those 3 billion letters, 99.9% of our DNA sequences, yours and mine, the sequence of letters looks the same. That means, in other words, that of those 3 billion letters, there are 3 million differences on average between any two humans. That's that 0.1% of average humans that's different. And it's those differences that make us unique, as I mentioned earlier. So that means that each of us are mutants. Every single human is genetically different from every other human. And that's not because of genetic modification. Those differences, those 3 million differences in the DNA sequence between each human, is a result of those random genetic changes that happen during cell division, when our chromosomes are being copied and our cells make rare mistakes in which letter they put in each spot on each chromosome. So you and I are very genetically similar in terms of when you look at percentile. You and I might be 7th cousins or 12th cousins. But when you look at the DNA sequence level, there are still a lot of differences, and that explains a number of the differences between each of us. Now, I also mentioned that I have wet earwax, and I was only mentioning that because that's another example of a naturally occurring genetic modification. Depending on where you're from in the world, this is another trait that's related to ancestry. You will either have, probably, wet earwax, or you'll have dry earwax. And geneticists have also discovered that people that have wet earwax at one spot on one chromosome, one single letter, if you have a G there, you tend to have wet earwax. But if you have an A there, you tend to have dry earwax. Again, this is probably a meaningless genetic difference, and it's probably not making one group of humans that have wet earwax better than people that have dry earwax. It's just a difference. But this is another example of a naturally occurring mutation that makes a change in a trait. And this is exactly what scientists and farmers do when they are trying to improve crops or livestock. Is we look for these differences in traits and then selectively breed for them. If you love cats, if you love dogs, if you love pigeons, if you love rats, you know that these are fanciers, people that breed for specific traits. And the traits that those are being bred for initially arose, they appeared, not evolved, they appeared because of DNA sequence changes. These random changes to the letters of the chromosomes give those organisms the different traits that farmers and scientists and so forth can set up crosses between and try to select for organisms that have those traits. So that's just the basics, important background information to understand a little bit more about how genetically modified organisms are going to be created. Every living thing has DNA. The sequences of their chromosomes are different, but it's those random changes in their chromosome sequences, the sequence of the A's, T's, G's, and C's, that change the proteins that those sequences encode, and those changes in the proteins then cause changes in traits that we can sometimes observe. So the fact that I have wet earwax is because I have a single particular letter at one spot on one of my chromosomes. The fact that I'm lactose tolerant means that I have a specific nucleotide, a specific letter, at one specific spot in the lactase gene on my chromosome 2. And so what a lot of genetic modification is about, as you might expect now, is choosing, artificially somewhat, which letters change to which other letters in a designed sort of way to make specific sorts of changes to organisms. And again, I'm going to try to leave it up to you to decide by the end of this presentation whether or not GMOs are good or bad. I suspect that you're going to find that there's a continuum and that some are good and some maybe are not so good. So as an example of that, here are just two different examples of mutations. I mentioned that not all mutations are bad. Some are. Some mutations, like mutations in a gene called BRCA1, which stands for breast cancer 1, mutations in that gene predispose people to getting breast cancer. So those mutations are certainly bad for the people that get them and their families. But we also started looking at this single gene change, this single letter change in lactase that allowed humans to be able to digest milk as adults. I would argue that that's a good mutation. You might not. That's fine. But these are just two examples. There's a spectrum of DNA sequence changes, mutations, and they all have different sorts of effects. So at the very least, I'm pleading with you, when you hear the word mutation, don't assume that it's necessarily bad. Because some are good, some are bad. So let's get at the really important part of this concept. What counts as genetic modification? As I mentioned earlier, I suspect that a lot of you may think that this image in the bottom left is a sort of representation of genetic modification, a scientist doing something artificial to a crop or livestock or a human, perhaps. On the other hand, I'm also going to argue that what you see on the bottom right, the artificial selection of traits in the ancestor of corn to produce what we now know as corn or maize, I'm going to argue that that's also genetic modification. Now, I hate, as an instructor, putting all text on slides, so I apologize. I'm not going to read this to you, but basically, I want to make the point that there is no consensus about what exactly a genetically modified organism is. There's no exhaustive definition that is all-encompassing. For example, the USDA, which you may or may not trust, depends, mentions the terms genetic engineering and traditional methods of producing improvements in plants or animals. And so that's an important distinction. Whether or not we're talking about genetic engineering, which is that scientist in the laboratory with DNA, they're also mentioning traditional methods like crop improvement, selective breeding for specific traits. And this, I think, is going to be a critical component of our exploration of GMOs. We could also look at the World Health Organization's definition, which is a little bit more specific in that they're talking about DNA alterations in a way that does not occur naturally. And that might be a definition that resonates with you. And that gets back, I think, to this idea of the scientist in the laboratory doing something. So we may think about these two definitions, and again, I invite you to think for yourself as we proceed through exploring GMOs, what is your personal definition of a genetically modified organism? Because there is not one legal definition even that we can fall back on. This term means different things to different people. And I expect you'll see as I give my presentation and you see what I think counts as a genetically modified organism. The examples I'm going to share with you today are humans, mosquitoes, and then we're going to look at a variety of different food crops that I think have been genetically modified, but maybe you disagree. So let's start with humans because that's probably the most controversial. Last year, in fact, the first genetically modified humans were reported to have been produced by a scientist in China. I want to point out that it's not necessarily that this was a Chinese scientist that this is important. In fact, I was really impressed that not only is human gene editing or genetic modification illegal in China, but the Chinese government actually stepped in and intervened with this scientist's work after he, by himself, announced that he had done this research sort of in secret. And he was eventually fired from the university he worked at. So responsible steps were taken after this came to light. But I just wanted to tell you briefly what he did, and you can again decide for yourself whether or not this was actually a good approach that this doctor took or not. Basically, the story is that each of our cells, or our bodies, produce a protein that the enzyme HIV, or the virus HIV binds to. So if this is a cell, we might have a protein that's on the outside of that cell that has a particular shape. And that shape is specifically recognized by that virus, HIV. And it's that physical interaction between the protein on the HIV and the protein on our cell that lets HIV grab onto the cell and get inside the cell and replicate and spread. So what this scientist wanted to do was genetically engineer a human so that that protein that's the HIV's sort of latch onto cells wasn't going to be put on the cell surface by these genetically modified humans. So that was his goal. And we don't know, the humans are still babies. There were 2 of them that were born that were supposedly genetically modified, not confirmed yet by scientists, but he supposedly genetically modified them for this beneficial purpose. The idea was that they would not be able to contract HIV. Single change, getting rid of one protein that is HIV's doorknob to get into our cells. So of course, one of the huge issues with anything genetically modified is an ethics question. Is it right to do these sorts of things, even if they're for a beneficial purpose? Because the main concern that most scientists and ethicists have, and maybe you as well, the public, are what if things don't work exactly as the scientist intends? So this was done for a presumably beneficial purpose. It may not have worked, and we don't know yet what the outcome is. And so that is definitely something that I agree with as a scientist, is that any sort of genetic modification that's done with genetic engineering, which is the scientist in the laboratory changing DNA sequences, that needs to be done with significant oversight and with small trials over long periods of time so that we know what the potential dangers or drawbacks or side effects are before things become standard or typical or normal. Now, on the other hand, this is a slippery slope because we already do some kinds, sort of genetic modification in the sense that we can select against human offspring that have certain traits already. Specifically, most pregnant women in their second trimester are offered a quad test that tests for 4 different substances that helps doctors identify whether an infant, whether a fetus, is likely to have Down syndrome, which is caused by having an extra copy of chromosome 21, another syndrome, which is caused by having an extra copy of chromosome 18, and then spina bifida and also abdominal wall defects. So these tests are given to let families prepare for the possibility that their baby would have one of these conditions. But of course, in states where abortion is legal, women then also have the opportunity, if they learn about this before the second trimester, to have an abortion based on what they might discover about the genetics of their baby. So this is not genetic modification again, but this is using genetic testing to select for particular mutations. Maybe someday we'll discover that there's a gene that controls nose shape, just choosing something at random. And what if someday we're not able to, or we're not allowed to, genetically modified humans to produce a specific nose shape, but we can select against fetuses that don't have that nose shape? Would that be the same thing, or at least have the same societal effect as genetically modifying? We can already do the former. We can already, not that we should, I'm not saying that that's the case, but we can already select for and against fetuses that have specific traits. So GMO is basically the next step down that slippery slope, in my mind. Now, aside from humans, a really well-advertised genetically modified organism before we get to crops is genetically modified mosquitoes. So here's another story where the intention is good, but are we moving too quickly as scientists to release these genetically modified mosquitoes? The idea with this story is that they want to release genetically modified mosquitoes that will mate with natural mosquitoes in the Florida Keys, and the genetic modification to these mosquitoes is that they will cause any of their offspring to be sterile. So the idea is you have a fertile male and you mate them with a fertile female, but one of those is a GMO. So for example, if they released fertile female mosquitoes into the wild and they found the fertile males to mate with, that genetically modified organism means that mosquitoes, their offspring, get produced, but they're sterile. So that means that the goal of this project is to try to eradicate mosquitoes in the Florida Keys. Because mosquitoes are a major carrier of human diseases, they want to reduce the mosquito population, and this is one way to do it. Not by spraying a pesticide that might be bad for more organisms than just mosquitoes, but by releasing specifically genetically modified mosquitoes that will make their offspring be sterile. That is, the next generation of mosquitoes won't be able to have kids of their own, and that would cause a decline in the mosquito population. So is it a good thing, or is it a bad thing? The human health aspect is definitely good, but what are possible side effects? What could go wrong? What sort of things could go wrong? So this is good for disease spread control, but a major concern about all genetically modified organisms that are released into nature is what happens, except for in this case, because they produce infertile offspring in theory, what happens if you have a genetically modified crop in a field next to a wild crop? And let's say the farmer wants to be able to sell their non-genetically modified crop as not genetically modified. Well, what happens if some pollen from the genetically modified crop floats over into the next field and pollinates the non-genetically modified crop? Well, now that formerly not genetically modified crop has some genetic modification in it. And so that is one of the biggest concerns about any sort of release into nature or even into farmland of genetically modified organisms, is how do we keep the genetically modified organisms separate from the non-genetically modified organisms? And that's a concern that a lot of people have, but I'm not sure it's founded in reality, and we'll maybe have time to talk about that sort of towards the end of the presentation. The short story for now is that genetically modified crops aren't themselves necessarily more or less healthy. Some are actually more healthy than others. There's no immediate health hazard to humans of genetically modified crops. The main concern, again, is the potential loss of natural crops if genetically modified organisms are released into nature or into agriculture. So keeping the wild, so to speak, crops safe and not contaminated with genetically modified organisms is the main concern. So that's one of the potential dangers, is, as I just mentioned, that first bullet point, what happens if the genetically modified organism spreads its DNA into natural populations? That's mainly an ecological concern. Another one is, what happens if the genetic modification that scientists intend have more effects than just the one that the scientist wanted to have? And that, again, is a reason why it's important to have lots of trials before things are released into nature or into the public, to see what sorts of effects specific DNA sequence changes have on the organisms. Now, I've talked a lot so far, and maybe you're starting to get a little bit tired of my voice, so I'm going to ask you to participate a little bit, if you'd like to, in the chat in just a minute. But let's go to something that's maybe a little bit more directly relevant to you, and talk about a few GMO foods. And I'm going to use these 5 different examples and a couple others, actually. We're going to talk about apples, rice, salmon, corn, and potatoes. So hopefully at least one of those 5 you personally identify with as either something you like to eat or something you don't like to eat or something you're concerned about. So the question here is, how would you know as a consumer and as a citizen, a member of society, not necessarily a US citizen, I don't care about that, but as a member of society, if you went into a field, or it was Halloween recently, so a pumpkin field, let's say, and you wanted to know, is that pumpkin genetically modified or not? How would you know? So here are a selection of pumpkins. They're kind of square shaped, so how would you know if that was a genetically modified pumpkin that caused them to form cubes instead of spheres? What sort of things would you look for? Well, in this case, this is actually an advertisement for a product called the melon mold. So no, these square pumpkins are not genetically modified pumpkins. These are pumpkins that are grown inside plexiglass or plastic containers that are cubes. So as the pumpkin grows, it fills the cube, sort of molds to its shape, and then the plastic cube is removed and you get a square pumpkin. But how would we know that if these sorts of things aren't labeled, for example? Should genetically modified organisms food or modified organisms be labeled as GMO? And we'll talk about that a little bit towards the end of the presentation as well. So in this case, this is not a genetic modification. But how do you know? So I think the way it makes sense to start is, well, let's think of what are any sorts of foods that we could eat that could be genetically modified. Should GMO foods be labeled? Is there anything you can think of? And if you'd like to, please type some ideas into your chat if you have that available. Otherwise, I'll produce a list myself in a minute or two. Some foods that are not containing DNA. So instead of what this slide says, I'd like to generate a quick list of foods, things that we eat that do not contain DNA. That is, foods that didn't come from a living organism. What things do we ingest that are not potentially genetically modifiable? I'll start with one. Salt. So table salt. It's a mineral. It's a rock. That never contained DNA, so you can't genetically modify our salt supply. So we've got sweet potatoes as a possible GMO food, so I'll list those separately. So actually I should have, so sweet potatoes, potentially GMO, those do have DNA. Sweet potatoes are plants. Plants are living things. They have DNA, so they could be genetically modified. The only things that we eat that don't contain DNA, margarine, a synthetic compound. I'm going to go with baking soda, baking powder. Those are chemicals, just like cream of tartar. Let's see, water. Water is not a living thing, so it doesn't have DNA. That's about it, actually. Everything else we consume, other than that short list, could potentially be genetically modified. A lot of students in my classes, when I ask them this question, say soda. Then I say, well, what's your soda sweetened with? Is it sweetened with natural sweeteners like sugar? Because sugar came from sugar canes, which are plants, which had DNA. Some students say beer. Well, yeah, but beer is fermented by yeast, and yeast are living organisms, so your beer has DNA in it. Same with wine. Wine came from grapes, and those grapes had yeast on them, and so on and so forth. So the point is, basically everything we eat, other than this short list and maybe a couple of other things, is potentially genetically modifiable because they all come from living things. And so that's why it was surprising to me, this has been a few years ago now, 2015, there was a poll in the Midwest that was put out, and they asked one of the questions to the people that responded was, do they support mandatory labels on foods containing DNA? And then as this title says, DNA. And that's the point that I'm trying to make is a lot of people think, and I don't disagree, but we're going to hear both sides of this story. A lot of people think that genetically modified organisms should be labeled. Maybe they should, maybe they shouldn't. But everything that has DNA in it that we eat maybe is GMO. We don't know. And so that's, I think, why 80% of Americans in this survey supported mandatory labeling on foods containing DNA, but that would be everything in the grocery store, except for some things in the baking aisle. So I think we have to be, and I think that's a good point that was made in the chat from Frank, that margarine probably still contains some plant products because it has to get its oil from somewhere. And if it's a plant oil, it may not contain any dairy fat, but it probably contains plant fat, so it may still contain DNA. So most of what we eat and buy at the grocery store and grow in our yards maybe has some genetic modification in its past. And so that helps us turn toward crops. All of these I personally consider to be examples of genetically modified organisms, but this is where you and I might start disagreeing if we don't already. So let's see. This is maybe the most controversial point that I'll make today. I think grapes are frankenplants, and citrus foods, and citrus too, because of grafting. A lot of people, well, not a lot of people, some vocal people call genetically modified organisms frankenfoods, after Frankenstein's monster. But really, if you think about it, a frankenfood would be a food that's cobbled together from a bunch of different organisms to produce something like Frankenstein's monster. And that's pretty much what we do with grapes and with citrus and with apples, is we graft. We take a part of a plant that produces a fruit that is a quality fruit that has the characteristics the grower wants. But those plants that produce really tasty fruit are usually not as susceptible to resisting pathogens, diseases. So what we typically do, citrus farmers, apple farmers, grape growers, is you will graft, you'll take the fruit, part of a plant from a plant that grows nice fruits, and you'll graft it onto a rootstock. That's the part that goes down into the soil and interacts with all the microbes in the soil, bacteria and so forth. And that rootstock is usually a rootstock that's resistant to pests. So we take the good qualities of two different organisms and we physically join them together, just like Frankenstein's monster. I think that's genetic modification. That's making one organism out of two different organisms that have two different traits. You may not agree with me, and that's fine. This is just an extreme example. But I think this maybe counts as a genetic modification. This doesn't, in my mind, and according to the World Health Organization definition, count as genetic engineering, but I think it's still genetic modification. So should those be labeled? Now, a great example of what I think, again, is genetic modification is traditional breeding or crop improvement. These are both wild tomatoes. This is more representative of an ancestral tomato, like a wild tomato, from centuries or millennia ago, arose in South America first. But then as these plants were grown in fields, occasionally a farmer might see a tomato plant that makes a slightly larger tomato. So that's what traditional breeding or crop improvement or selective breeding is all about. You find the really rare plant that makes a slightly better fruit. Better could mean anything. In this case, I'm talking about size. It could be sugar content or antioxidant content, whatever. And you grow the seeds from that plant to try to get more plants that make the bigger fruit. And then eventually, after maybe generations of growing, these plants, these lineages of plants, you find another tomato that's even slightly bigger. So you pick those seeds and you use those to make more plants that grow bigger tomatoes. And so eventually we get to things like globe tomatoes. These look nothing like the wild tomatoes. I mean, they're the same species, they just make huge fruits. And that's because farmers, people that were growing the plants, selected for that trait. Every time they found a mutant, one of these plants has maybe a few genetic changes that change the proteins in their cells, and those changes in the proteins cause the fruits to grow larger. They select for the plants that have those mutations that were naturally occurring during DNA copying inside the tomatoes as they were propagated in the fields. So that's not genetic engineering, where a scientist specifically made a change to DNA sequence, A's, T's, G's, and C's, to cause this difference. But nevertheless, this difference is caused by mutations, genetic changes that distinguish this plant or this tomato from this tomato, or the types of naturally occurring genetic changes that make you and I different. Now here's a true example of a genetically modified or a genetically engineered organism. This is a salmon that's been sold in Canada. It was approved for human consumption in the US, and I believe it is being sold today, or at least has been sold in the US. These are farmed salmon, so they're not wild-caught salmon. They're farmed usually in open water, floating netted pens in the ocean, sometimes in freshwater, depends on the species. And one of the concerns about farmed fish is they take a long time to grow to adulthood, so it's expensive for the farmers, and so it's expensive to us, the consumers. So the goal here was to make fish that grow faster. So consider this. Do you think to yourself, once I tell you this story, is this harmful genetic modification or not? What the scientists did was they engineered the salmon just to make more of their own growth hormone protein. So this is not Frankenstein's monster taking growth hormones from other species and putting them into salmon. This is just turning up the amount of production of the protein, the hormone, that salmon already make naturally themselves. The scientists just changed a couple of letters in the DNA code that make the salmon produce more of that naturally occurring growth hormone, so they grow faster. That was genetic engineering. Scientists specifically made those changes to the salmon's natural chromosomes. But the change itself, the increased production of this growth hormone protein by the salmon, that could have happened naturally. That change in the DNA sequence might eventually, given enough time and enough salmon, have happened in nature. And maybe it has happened in nature, but we never saw it happen, because people weren't looking at that specific mutant fish and farming it and trying to sell it to somebody. So is this a good GMO or a bad GMO? There are probably pros and cons. And I'm not here to answer that question for you. That's a question that, again, you'll have to answer for yourself. Here's one of my favorite examples. Arctic apples. Apples typically brown when you cut them. That's because this protein, an enzyme called polyphenol oxidase, when you cut open an apple cell, that protein spills out of the cells, and that protein reacts with oxygen to cause the fruit to start to brown. So, if you want to make fruit that doesn't brown when you cut it, what do? You genetically engineer those apples to not make as much of that protein. So again, here's a case of genetic engineering. Scientists artificially turned down the amount of this enzyme that apples produce, so that when you cut the Arctic apple, I think there probably should be a trademark or maybe a registered, I'm not sure, the company owns them, they don't brown as quickly. So here's a traditional apple in comparison with an Arctic apple. McDonald's probably loves this. Those pre-sliced apples in the kiddie meals. I'm not picking on McDonald's specifically, but that's the point, is the grocery store packaged pre-sliced apples have a longer shelf life based on this one change. And again, I would ask you, this change, the reduction in the amount of one protein being produced, can happen naturally by chance when that gene sequence of that protein undergoes those random mutations that happen very rarely, but naturally, when cells divide as organisms grow. In other words, I would argue that somewhere in the world, in some apple orchard somewhere, there are enough apples that this mutation that was introduced artificially in the laboratory has also happened in nature. But because a farmer was probably not really closely paying attention, they would have to slice open all of the apples in all of the orchards to find the ones that brown less. We just haven't seen that mutation happen in nature. So a scientist just accelerated the process and did it in a laboratory. So which do you think is better? They're the same mutation. This is a thought experiment, of course. This isn't true, but it would be the same mutation. Is it problematic that a scientist does it in a laboratory more quickly and more efficiently than we would have to wait to observe that same mutant organism in an orchard somewhere in the world? And I think the distinction comes down then not to the fact that the DNA sequence changed, because that can happen artificially by genetic engineering, or naturally through the natural process of mutation that happens as organisms reproduce and grow. I think the main difference is an ethical one. Do people feel comfortable with the idea of scientists genetically engineering in a laboratory? And that may be the root of the conversation. Now again, I don't know anything about the population that's in the audience, but I'm guessing that a lot of you either love or rely on corn or rice as a major staple. And so the last couple of examples I'm going to give are the production of corn and a genetically modified rice strain. So here's the story of the evolution of corn. Corn, a couple thousand years ago, looked like this on the left. It was a tiny little weedy plant called teosinte that grows in Mexico. And it's not a very good food source because the nutritious part of the kernels are underneath this really thick, almost impenetrable seed case. And of course, this on the right is what we know of as maize or corn today. And in between is a hybrid between teosinte and maize. So this story is all about how did we get from the ancestor of modern corn to what we know of today as maize or corn. And what are the genetics of this change? So here's a gentleman on the left with a teosinte plant. Again, that's the ancestor. That's what the ancestor of Fresno State's Gibson Farm Market sweet corn looked like, and still does today. And that is that plant that produces the teosinte-like fruit. Not very nutritious, really hard for humans to get into and get access to. And if you can break into that tough sucker, you destroy the fruit at the same time, so it's hard to get any nutrition out of that really closed teosinte fruit once you actually manage to break it open, if you do. So, archaeologists and anthropologists know that the domestication of corn, what we think of as corn, happened in central or southern Mexico. And this was published about a decade ago in a scientific journal, this study, looking at the origins of domestication of corn. And it happened, at least one origin of, or the area where corn was domesticated was near this shelter, this giant boulder where there's a space underneath the boulder where humans would use that as shelter. And these scientists found tools that were used for grinding or milling corn, and those were dated about 8,700 years ago. These were being used by natives in central Mexico. And they know that that was about the time when teosinte started to be domesticated into corn, because they were able to look at microscopic chunks of plant material that are embedded in these rocks, and they found plant material that looked more like corn kernels than teosinte kernels. So that's how they figured that about 9,000 years ago, corn started being domesticated in central Mexico. Now here's where we get back to genetic modification, and specifically DNA sequence changes or mutations. On the right, or on the left here, again, the individual labeled B in this figure is one of those ancestral teosinte with the closed fruit cups. Really tough shell, essentially, outside of the fruit. And what these scientists did was they mated that ancestral teosinte plant with a corn plant, a current corn plant. And they discovered that their offspring has these open cups. The fruit is much more accessible to humans to access and to use as a food source. These scientists discovered, like the story about earwax and like the story about lactose tolerant, there is a single letter change in the DNA sequence that converts teosinte to something like that hybrid. That is, there's a point mutation, a single nucleotide change that makes those really tightly closed fruit cups open up and accessible to humans to more easily extract as food. So that's the story of the domestication of corn. It's all based on a naturally occurring mutation, a single change in the DNA code of teosinte that made farmers able to start selectively breeding for bigger plants, larger fruits, but it all started with being able to have access to those fruits. So is that genetic modification? It's traditional breeding, but it's based on a single DNA change. So would it be different if a scientist in the laboratory made that single DNA change? Does it matter if the mutation happens at random in nature, or does it matter that a scientist produces that single DNA sequence change in a laboratory? Now I'll give you a couple other examples of crops, and I'll wrap up hopefully in about 10 minutes or so. We'll have some time for Q&A. You may have heard of Bt crops, and these are crops that are definitely genetically engineered. This is, of everything I've talked about so far, this is perhaps one of the more famous and maybe controversial types of genetic modification or genetic engineering. This is a case where a gene from one species has been put into a different species. Before we were talking about things like modifying the amount of enzymes that an apple creates to reduce the amount of browning. We were talking about turning up growth hormone production in salmon to make them grow faster. This is a frankenplant, and this is the concept of a Bt crop. There's a soil bacterium called Bacillus thuringiensis, which is where Bt comes from. And those bacilli, those bacteria, produce a toxin. The good news is the toxin is not toxic to animals, like humans, but it is toxic to other animals that might be pests, like the corn borer. So scientists have taken the gene, the Bt gene that produces that toxin, and they put that gene in the plant so that now the plants can produce this bacterial toxin. So when pests, like the corn borer, eat the plant material, they are affected by the toxin and it kills the pests. And again, this toxin, the Bt toxin, has been shown not to be toxic to humans. So it has no effect on us. It has an effect on crop pests. That is definitely genetic engineering, and maybe that is something that should be labeled. So again, I'll leave it to you to decide whether or not that is an example of a GMO, and whether that is an example that maybe should involve some sort of labeling or notification to the public. And it's easy to tell if a crop has Bt in it. So that is a form of labeling that a lot of companies do perform. Another example, golden rice. This is one of those maybe intermediate examples, where we know that across the world, a lot of children have a deficiency in the amount of vitamin A that they consume. And this leads to hundreds of thousands of deaths per year across the world of children less than the age of 6, and maybe 500,000 cases or so annually in kids worldwide having blindness, permanent blindness, because they have a vitamin A deficiency. So what some scientists did was they added 3 genes. There are only 3 DNA sequences that produce three proteins that allow the production of beta carotene, which is a nutrient and it's a precursor of vitamin A. These three genes, importantly, are already in rice, but those three genes only produce their proteins, the three proteins that are encoded by those genes, in the leaves of the plant, not in the fruit of the plant. So while these rice plants, all of the cells, have those three genes aren't active in the kernels of the rice. So they don't put the vitamin A in the rice kernels, they put the vitamin A in the leaves of the plants, because this is part of photosynthesis. So that's all these scientists did, was they took genes that are already in the rice, but they found a way to turn on those three genes to produce beta carotene in the rice, and that's why the rice looks different and it's called golden rice, because it's potentially more nutritious in terms of its vitamin A content than typical white rice. So a good intention, and it's genetically modified, but it's just turning up the amount of genes and where in the organism these genes are produced. Rice that produce those three proteins encoded by those three genes in the rice, not just in their leaves. So I'm going to end with a couple other quick examples so that we can get to Q&A. This is what we've talked about so far. Traditional breeding, you may or may not think of as genetic modification. I include it in my definition. You don't have to. We talked about corn and we talked about tomatoes and selective breeding. We talked about the concept of grafting, which produces a frankenplant, but I think most of us consider that that's an okay practice. And then we've closed with talking about genetic engineering, where scientists did specific things to the DNA of organisms. We talked about mosquitoes, humans, salmon, rice, and Arctic apples. Whew. So that's a lot of GMO. So the takeaway, I think, if I have anything as a geneticist to share with you, is again this thought that I've already brought up, which is that genetically modified organisms sometimes do things that would happen in nature. So if we remember back to the World Health Organization definition of genetic modification, it counted things that weren't natural. But again, the mutation that causes apples not to brown could happen in nature, and maybe it did, maybe we just didn't spot it when it happened. So is that an important thing to consider when we're talking about whether or not genetic modified organisms are useful or not, whether they should be labeled or not? I want to end with a couple of examples, including glyphosate. I saw glyphosate just come up in the chat, so we're going to talk about that briefly after this example. So I'm going to close with two examples of ways that things might be unhealthy when they're genetically modified. So I've talked a lot about the good, and I've had some caveats about, well, maybe this would be bad. So here are two examples, because I'm an academic and I believe in representing both sides of the argument, two examples where things might not have gone the way that the scientists intended. This is a toxin. This is the chemical structure of a toxin called solanine, and it's produced by plants that are in the family Solanaceae, which is the nightshade family. So you may, I don't know who's in the audience, but you may have been told by your parents not to eat the deadly nightshade. It's called deadly nightshade for a reason. And lots of foods that we eat are in the nightshade family, like tomatoes and potatoes. They don't produce very much of solanine, so they're safe for us to eat. But you may also have heard at some point, it depends on where you come from and who your grandparents were maybe, not to eat green potatoes, because green potatoes, the green spots on potatoes might be places where they're starting to produce more of that toxin. So you have to be careful with plants in the Solanaceae family, the nightshade family of plants. But this story is about potatoes, part of this plant family. And you probably know that potatoes, there are lots of different types of potatoes. If you go into the grocery store, and you can get potatoes that are good for boiling, good for turning into French fries, turning into potato chips. And different varieties of potatoes have different qualities when you treat them in different ways. So effort to make a better potato chip potato, farmers had been selectively breeding for those qualities. They crisp nicely, they turn the right golden brown color when you fry them. And it turns out that one of the varieties of potatoes they produced, called the Lenape potato, and I know you can't see the details in this figure, I apologize, but that's not the point. The point is that in this study, published in 1980, scientists looked at the amount of solanine, that toxin, in different potato varieties. And look at what happened. I'll explain it to you since you probably can't see it in detail. Look at what the solanine production in the Lenape variety of potato, this perfect chip frying potato, is. It's 12.51, which is about 4 to 5 times more of this toxin in this potato variety than all the other potato varieties they looked at. Short story is that during traditional crop improvement, traditional breeding, farmers had been breeding these crops in the fields for decades, selecting for these potatoes that make this really nice, crispy potato chip. They accidentally also selected for a different trait. They produced great chip frying potatoes that would be toxic because they also happen to produce a lot more solanine. The point here is that just because something is genetically modified doesn't mean it's bad. And just because something is traditionally bred through crop improvement doesn't mean it's good. It's a good thing that this potato variety didn't make it into the public food chain. So even taking years to try to create better crops doesn't necessarily lead to a better crop. Now, last thing before we open up to Q&A. I know I'm a little bit over time before I start taking questions. Glyphosate, or Roundup, showed up in the chat. So let me tell you a quick story, one last story about glyphosate. It's a herbicide you might know as Roundup. Glyphosate is the active ingredient in the common herbicide Roundup, the thing that you maybe spray on your weeds to try to kill them. The way that glyphosate works is that that chemical binds to and deactivates a protein that plants make that's an essential protein for them to live. And it's abbreviated, that gene or protein is abbreviated EPSP. That's not important. So what people have done to try to allow them to spray their fields with glyphosate or Roundup and only kill the weeds and not the crops, is they've turned up the amount, they've increased, in other words, the amount of this protein that the crop plants produce. So the argument is, well, if the crops produce a lot more of this protein, and then somebody flies over and sprays glyphosate, maybe the amount of glyphosate, the herbicide, that they add won't be enough to deactivate all of that protein that these super plants are now producing, but it will be enough Roundup to kill anything that's not genetically modified, that produces less EPSP, like the weeds that are in the fields. So the goal here was to produce rice that make a lot of this protein, EPSP, enough that Roundup still wouldn't kill those rice plants when they're sprayed, but it should kill all the weeds around them. Of fate, it turned out that even if you didn't spray Roundup on these genetically modified rice plants, there was actually a benefit to those genetically modified rice plants. They actually grew faster than non-genetically modified rice. And that was shocking to scientists because one of our basic concepts is the concept of a trade-off, that there's a balance. And what the scientists had assumed was going to be the case is that these genetically modified rice plants would never be able to escape or to crossbreed because they have a disadvantage. They thought they would have a disadvantage if people didn't spray Roundup, because these genetically modified rice plants are using a lot of extracellular energy, nutrients, to make all of these extra proteins to be Roundup resistant. So they assumed that if they didn't apply Roundup, then those genetically modified rice plants would actually be less healthy because they were using all their energy producing this protein that they didn't really need to spend time and energy making. So they assumed that these genetically modified plants wouldn't be able to spread into natural populations. In an interesting twist, so the last story then is that that was actually found not to be the case. That even in the absence of this weed killer, glyphosate, Roundup, those genetically modified plants were actually still healthier than the natural rice plants in terms of their growth rate. So we've reached the end. Currently in the US, it's optional to label genetically modified foods, much like it's optional to label whether something's organic, and there are lots of definitions of what organic means too. That's not regulated either. Much like beef grading, grade A beef doesn't really mean anything. It's an optional program that ranchers can participate in. But I also want to mention, as a closure, that the American Medical Association and the American Association for the Advancement of Science have both suggested that labeling of GMO foods before we have any evidence that there's a potential health hazard could actually be misleading. Because if a consumer went into a store and they saw some things labeled as non-GMO and other things labeled as GMO, there could be this perception that GMO must be better. But that might not actually be the case. The jury's still out. So these are the questions, again, that I ask you to consider as we go back into the world and interact with GMO and non-GMO organisms. Should we genetically modify, and if so, what controls and restrictions should be in place? So, as a closing thought, knowing what you now know about DNA and about GMOs, is it reasonable for a scientist in a laboratory, knowing that the single nucleotide change, a G in teosinte at one spot on one chromosome versus a C, out of the billions of nucleotides, the billions of letters in teosinte and corn, if that one DNA change is so important, is it okay to do that in a laboratory? Or do we really need to wait for hundreds and thousands of years for somebody to happen to discover that mutant plant in the wild to be able to take advantage of this as a food source? So again, here is my contact information. If you'd like to email me, that's my email address, and again, that's my YouTube channel in case you want to watch this video in the future or other videos. And I'm certainly happy to stop now and to turn back to the chat and the Q&A and answer some questions. And thank you very much for showing up and participating. I hope this was meaningful to you. I hope you got something out of it, and I hope you will share what you've heard today with your family and your friends.
And Danny will help you get through the questions.